EDU-ARCTIC 2, Polar Research for Education, innovative program in Poland and Norway. Webinars.
In the framework of this project, we produce and propose some educational toolkits, which are focused on Arctic topics mainly. And educational toolkits are available in English, also in Polish and Norwegian, at least some of them. But they're all available in English on our website, and they're not overloaded with lots of information and knowledge, at least not usually. But they really aim at bringing the knowledge in a different way, with lots of different tools and online educational tools, videos, quiz, and so on. So this is the main goal, and those webinars that we organize accompany those educational toolkits, so they can serve as a wrap-up or as an introduction. So eventually all the recordings will be on our channel. And one of the topics is ecotoxicology. At first, it might not seem so much of an Arctic issue, but it's on the contrary, and we'll focus on Arctic regions. But first, we're going to start with ecotoxicology in general. So first, if we talk about ecotoxicology, we need to understand what toxic means. Anyone can name a lot of poisons which are dangerous, which are well known as dangerous to people, to humans, to animals and plants in environment in some cases. But actually not all of us understand completely what toxicity means. We only perceive well-known poisons as something dangerous. So what is toxicity? It's a potential of any kind of substance, of a chemical substance, to cause some effect, an adverse effect, a negative effect in a living organism. So it's a sort of a degree to which it can damage an organism. It can refer to a whole organism, it can lead to death, it can affect some sort of substructure, some sort of internal system, organs, or even cells. So it can refer to, something can be toxic to animals, bacteria, plants, and it can cause very different adverse effects in many ways, with many mechanisms. Nearly 500 years ago, a Swiss physician was presented here on this image. He was a physician, he was a chemist, and he is known as Paracelsus. He expressed the basic principle of toxicology, which is usually repeated as all things are poison and nothing is without poison. So only the dose makes a thing not a poison, or only a dose makes a thing a poison. And this is the short version that most people know. The dose makes the poison. So you can see it also in Latin here. So dosis facit venenum. So it means that a substance that contains some properties that can be dangerous can cause harm only if it occurs in a concentration that is high enough, or amount that's high enough. In other words, any chemical, even water, even oxygen, so chemicals that we normally do not associate with poisonous properties, can be toxic if too much is ingested or absorbed into the body. Too much in some limited period of time, of course. So the toxicity of a specific substance depends on a variety of factors, For example, how much of the substance a person or an organism in general is exposed to, how they're exposed, for how long. But actually, we tend to fear the chemistry, the awful chemistry, awful chemicals. We turn to more natural fabrics, natural substances, natural food. But the thing is that those horrible chemicals are exactly everywhere. So the air we breathe is composed of chemicals: nitrogen, oxygen, argon, carbon dioxide. We brush our teeth with toothpaste, and it contains some chemicals like fluoride and sodium carbonate. At breakfast, we might drink an orange juice, coffee, so they also contain some naturally occurring chemical ingredients: acrylamide, fructose, aldehyde. So even water, as I mentioned, the most abundant natural resource on Earth, even clean water, not polluted water, is a chemical substance because it's made up of two elements of the periodic table, which is hydrogen and oxygen. So also all natural products without any artificial aromas, without any preserving chemicals, they still, if described chemically, can look really frightening if we do not understand what we see. So there is also methylparaben in blueberries, oxalic acid in bananas. So lots of names that could scare us and think that they are poisonous. And as we know already, it only depends on the amount that would be consumed. And toxins, especially toxins that we really identify as toxins. That means that not a very huge amount needs to be consumed to have the adverse effects. So they have different mechanisms of causing the adverse effects. So some, like hemotoxins, are carried in the bloodstream and target red blood cells mostly. Others, like necrotoxins, of course, we talk about humans and animals, not plants. So others are very, very dangerous. Necrotoxins are particularly dangerous. They usually have very fast effect and are really, sometimes they're quite hard to identify, to find, and to counteract. So necrotoxins can cause tissue destruction because of cell death and also are carried in the bloodstream. But the most dangerous and the fastest to act and to cause adverse effects are usually the neurotoxins. Neurotoxins affect, of course, the nervous system. What we can identify one of the most poisonous objects, I would say, or because we do not know the name of actual toxin, but we know that some mushrooms are not edible, not only not edible, but they're very dangerous, and most of them contain not only but mostly neurotoxins. Also, we have now cytotoxins, and their mechanism is killing individual cells, or they're substances that are toxic at the level of the cell, not an organ or a system or organism as a whole. So how is toxicity measured? Because if we need to identify if something is really poisonous, is dangerous, or in what amount is it dangerous, it means that we have to calculate it somehow and have some sort of measure. So the measure that is used, actually there are two measures used, LD50 and LC. What does it mean? So LD50 is the lethal dose, which causes death of 50% of tested animals, and the animals are usually mice. So it's there is an experiment, and if half of the population exposed to toxin die, it means the lethal dose is. And it's measured in milligrams per kilogram or gram, usually kilogram of body weight. So it means that each, in this case, we measure that on the level of individual organism, but on the other hand, also on the population level. So each organism receives a particular dose, but not all the organisms react the same. So we have to find the moment, we would say, when half of them die. And the other parameter is LC50, which is lethal concentration. And this time it's an amount of substance in the air or in the water that also kills 50% of this population. But this time it's measured in parts per million, because we don't actually know, we might find out later, but for this measure, we're not particularly interested in the beginning how much toxin was ingested in the first place by each and every individual, because they could have consumed very different amounts of toxin. But we release some calculated amount of toxin into environment, which is into the air, into the water, and check which concentration causes death of 50% of this population. So these experiments are quite rough, and they inadvertently they just cause death of the animals. So they really need to be justified, but they are justified if we try to measure toxicity of new substance, of new combination, of new product. I mentioned water or toothpaste or blueberries. They can could all be toxins or toxic or even lethal, deadly, if consumed at once. But you'd have to consume a lot. But there are also toxins that are quite well known, not always because of their immediate effect. Because immediate effect is not always the case. Well, there are substances that cause immediate effect. Cyanide. It takes usually seconds to minutes to cause death of an individual after ingestion. But these toxins are also quite famous, mostly because of their environmental effect, and this is when we approach the Arctic region as well. So these three substances are DDT, PCBs, well, it's a group of substances, PCBs, polychlorinated biphenyls, and mercury. So starting with mercury, it's especially harmful not in the inorganic form that we may know from old thermometers. Some people still have them at home. They were very accurate. Now it's forbidden to sell them, at least in European Union, because of the fear that they would be easily broken and the mercury can be released into the environment. But actually, such cases were quite rare and not that deadly, because this is an inorganic form. But the most important from the toxicity point of view is metal mercury, which is organic form of the mercury. As for PCBs, they're organic chemicals. They seem quite to have quite simple structure, like carbon, hydrogen, chlorine atoms. So it doesn't seem to be that complicated. But actually, they're very complicated, and they do not break down easily once they arrive in the environment. Therefore, they can remain in the environment for a long time, and this cycle between air, water, soil. In the very beginning, I also mentioned DDT, a very famous or infamous insect killer, used to control the mosquitoes carrying malaria. And also, it's also quite durable, mostly because it, once consumed, it remains in the fat of the animals, and it may remain there for many, many years. So these are the characteristics. Actually, you can see that in the very beginning, DDT was advertised as something good, as a breakthrough, actually, and the inventor, or I should say reinventor of DDT, he was awarded a Nobel Prize, and now DDT is as a symbol of doing harm to the environment in the long term. It was very, very successful in controlling mosquito population, but it wasn't well tested, apparently, because the mosquitoes died, but the adverse effects were noticeable. That's the first thing. Maybe not on humans; they much less noticeable with humans, but they can affect hormone production, and also they affect fertility in animals, so they can endanger populations. But they are DDT is particularly dangerous for birds or egg-laying animals, actually, because they affect the egg shell thickness. So when there is an increase of DDT concentration in the ecosystem, it means that there is the eggshells are not that thick. And also it remains in the environment for quite a lot of time. On the left, you can see the half-life time in particular conditions, because it depends heavily on what conditions there are, whether it's in soil or in water. Then we have our mercury and a Mad Hatter, a photo of Mad Hatter from the movie Alice in Wonderland, and it's not a coincidence, because Mad Hatter actually is a character based on hatters in Great Britain and England, people who produced hats, who made hats, and in the process mercury was used, and many of them suffered from some psychological and mental problems. They were not always very serious, but they could be very serious. So mercury affected nervous system, central nervous system. So the Mad Hatter wasn't just didn't end up in Alice in Wonderland coincidentally. There were a lot of, maybe you wouldn't say mad hatters, but people with a lot of problems with nervous system because of ingestion of mercury, of being exposed to mercury while making hats. And DDT was actually banned in the US in 1972. So in the 30s, like at the end of the 30s, it became very popular. And then in the 70s in the US, it was totally banned. And as for PCBs, they were synthesized quite a long time ago, in 1929. And they have a large or wide range of toxicity. Now they're no longer commercially produced, but they still remain in the environment. They can be still present in products and materials that were produced before the ban, because PCBs were also banned in 1979. But they can still be found in the product and also in the environment. So what are the mechanisms behind those toxins being present in the environment for such a long time? And why is it a problem? And why is it a problem especially in regions such as the Arctic? Here you have two environmental cycles or processes presented. They're more like processes. So bioaccumulation and biomagnification. The first one refers to one organism or a group of organisms, but the level of toxin product increases within the same organism with time. So with time, for example, fish consume more and more toxic algae, polluted water is ingested for an increased period of time, and in the end, the level is really, really high. But it all happens within one organism, or is observed within one organism. And then we have biomagnification, and biomagnification is a little bit different. It also is based on increasing level of toxin in time, but this time it's not only the time that matters, but also the increase between trophic levels. So in the very beginning, there are some toxins, for example, zooplankton, which is then later consumed by fish, and with each and every level, the amount of the toxin increases in another higher trophic level, up to apex predator. So that's basically the main difference. You can see it explained here. So this in bioaccumulation, in general, the result is that the concentration of the toxin in the organism is higher than in the surrounding environment. And the uptake of toxin can take place via respiration, food intake, some epidermal skin contact, and there is a gradual build. And actually, bioaccumulation is a very natural process, allowing us to survive, not only us, but all the living organisms, because organisms accumulate essential nutrients, vitamins. So we need to accumulate. Unfortunately, we also accumulate toxins. So there is a buildup in the tissues so that we can grow, so we can regenerate our tissues. And so it is absorbed faster than it is removed. So this is bioaccumulation once again. And here in biomagnification, we have different levels and the tendency of pollutants to concentrate as they move from one organism to another by being consumed by this another organism, by this higher trophic level. One of the conditions to make biomagnification possible is that pollutants are durable and they are not metabolized. So because if they are metabolized, they would be they would leave the organism before they could be transferred to another trophic level. But in this case, they're not metabolized, so the organism containing pollutant is eaten and they're passed on predator. But these two processes still have something in common. So in both cases, toxins are ingested, and bioaccumulation actually can lead to biomagnification. And there is a time factor, so the concentration gets higher and higher over time. Like I said, biomagnification is not always possible. So when something can be diluted, broken down, excreted in urine, for example, this substance cannot be bioaccumulated. But in something that's dissolved in fats, like I said, the DDT remains in fat tissue because there are no enzymes that degrade the substance, that means that this is a persistent pollutant and it can allow biomagnification. But how is it possible that we're even talking about toxicity in the Arctic? The Arctic is quite large. There are some human habitats, but in general, in comparison with other regions, we could say that at least in some parts, we would expect a very, very clean environment, cleaner than anywhere else, with no big industry. Most of the mines don't work for now. I mean, coal mines in some regions. So in general, there is less intense transport, less cars, lots of empty space, and really simple living. So how do the toxins reach the Arctic? And they do reach the Arctic. And the ice, we know that from, for example, ice core samples from Greenland inland ice sheet. And we know that industrial pollution has had an effect on the Arctic region for about a century now. And one of the factors is so-called grasshopper effect, which sounds really nice, but really isn't from the Arctic perspective. Also known as global distillation. So it's a phenomenon which is seasonal in its nature, which causes basically toxins to jump from the source to remote locations through successive evaporation and deposition. And this is how transportation is enabled, also following atmospheric currents. So in the Arctic, it does happen. So toxins jump, and it depends from where they jump, and therefore what kind of toxin depends on the season, thus seasonality. So the east and the west coast of North America is a source of this pollution in winter, which are big cities, industrial regions. And in the summer, it's mostly from Central Europe and also power station from former Soviet bloc. So this is how these remote sources cause pollution in the Arctic, which is quite high, surprisingly high. When I was talking about bioaccumulation, biomagnification, I used the example of fish. And it was not a coincidence, because, for example, in the Arctic region, the Arctic Ocean, northern seas in general, are great fishery grounds. So they're the source of lots and lots of fish. So if there's a lot of pollution, how come are we told to eat fish? So we're being told that eating fish is healthy in general, that fish have a lot of nutrients. But on the other hand, if they accumulate, they uptake so much toxins from the surrounding environment, from the water. Should we eat them, really? Well, there are some studies, and this is a study from 2013, focusing on mercury levels, because mercury is mostly this toxin that is a toxin of concern and is stored in fish body, which is then later consumed by humans. So the scientists identified fish with highest level of mercury, which was king mackerel, swordfish. Well, maybe not that popular in most countries. So, tilefish, less popular, and shark. I think it's quite rarely consumed, but king mackerel and swordfish is actually consumed. And these are fish with a highest level of mercury. And as for lowest, therefore safe mercury levels, there's fish, but there's also seafood. So we have carp, trout, crab, scallop, that's seafood, and and so on. So in 2013, these are the findings that eating fish can be dangerous if you eat a lot of fish because of mercury load, but some species have higher levels than others. When we're talking about fish and mercury poisoning, actually, it's not possible to avoid talking about Minamata disease. It's actually the most famous case of food poisoning, I would say, especially methylmercury food poisoning. Sometimes it's referred to as Minamata disease, and it's a neurological syndrome caused by severe mercury poisoning. And symptoms are quite horrible. So they include ataxia, first numbness in hands and feet, general muscle weakness, narrowing of the field of vision, damage of hearing, damage of speech. In extreme cases, very serious mental problems, paralysis, coma, and even death following a couple of weeks of the onset of symptoms. So it is very dangerous and it has horrible effects. And it was first discovered in Minamata City in Japan, in Kumamoto Prefecture. And it was in 1956. This is where or when the first patient was officially noted. And it was caused, now we know that the outbreak of this disease was caused by the release of methylmercury in the industrial wastewater. So the source of pollution was this Kiso Corporation's chemical factory. It continued releasing this wastewater from 1932 to 1968. And this highly toxic chemical, methylmercury, bioaccumulated in shellfish and fish in Minamata Bay, and the local population ate a lot of fish. They were fishermen, and it resulted in mercury poisoning. But they also fed fish to dogs, pigs. So the deaths of cat, dogs, pigs, humans continued for over than 30 years. And the government, the experts, and the company did little to prevent the catastrophe. They didn't even want to admit that they were causing this pollution. And as for the beginning of 21st century, March 2001, almost 3,000 victims had been officially recognized, of whom almost 2,000 died. And some people had more minor effects in comparison with those really serious cases. In the end, more than 10,000 people had received financial compensation from the company, who finally had to admit they were releasing this methylmercury into the water. They paid like $86 million in compensation, and they were also ordered to clean up the contamination. So this is considered the big pollution disease with very serious effects, and the consequence of not testing enough what it would bring into the environment, and being quite reckless actually with what we're releasing it, and not admitting there's a problem. So as you can see here, and kind of the Arctic, one of the problems while bioaccumulation and biomagnification is a problem here. First is because there is a lot of poison or toxic materials which are durable or brought to the Arctic, amongst others, grasshopper effect of global distillation. And on the other hand, we can see that the trophic, the food web in the Arctic is quite simple, and by simple I mean it is all based on the algae and then phytoplankton, then zooplankton, more zooplankton. It can be complicated between the links, but there is not a lot of choice of food for the links of this food web. So you cannot switch to another type of food. And if this basic source of food is contaminated, then it causes everything to collapse. And with biomagnification, we know that the levels of contamination increase from one trophic level to another up to the apex predator, which is polar bear in this case. Actually, now there's some research saying that the levels of mercury in the fur of polar bears decreased actually, but it's not that good news, meaning that it doesn't mean that there's less pollution or less mercury in the environment, or that the environment is cleaner and the animals are safer. It actually means that because polar bears are very short of food, so they can, because of the sea ice decrease, they cannot hunt seals that they would normally rely upon. They do not consume as much mercury, so they avoid this. The apex predators is avoiding this trap of the Arctic simple food web. But on the other hand, it means they need to switch to bird eggs, plants even, and it does not provide them as much energy as they need. So even though they avoid this contact with mercury, it only means that they have to switch to another food source, which can also lead to their extinction. So it's not always that the result of research saying there is less mercury can be interpreted that it's good news because the environment is less polluted. Sometimes it's more complicated than that. So that's it for today. That's all I wanted to talk about.
Watch other recordings from webinars on our YouTube channel, youtube.com/EDU-ARCTIC. EDU-ARCTIC 2, from polar research to scientific passion, innovative nature education in Poland and Norway receives a grant of 240,000 euros received from Iceland, Liechtenstein and Norway under EEA funds.